All right. Thanks everyone for coming out today. My name is Matt Nelson. I'm a senior research scientist here at the Field Museum. And before we get started, I just wanted to acknowledge that the Field Museum resides within the traditional homelands of the Three Fires Confederacy, the Ojibwe, Odawa, and Potawatomi. And I wanted to recognize and express my gratitude for the original peoples who laid the foundation for the city of Chicago and for the diverse indigenous nations that reside here today. So it's my great pleasure today to introduce Dr. Daniel Stanton to the Field Museum. Daniel's an assistant professor in the Department of Ecology and Evolution and Behavior at the University of Minnesota Twin Cities. And Daniel's a collaborator and someone I've known in varying capacities for about 20 years. We first met when he was an undergrad working on fungal pathogens of water fleas at the University of Wisconsin-Madison, where he ended up majoring in botany and biochemistry. He then went on to conduct his PhD work at Princeton, where he worked to better understand the roles of epiphytes in fog desert communities. Following this, he had a postdoc at the Australian National University and then worked in varying capacities at the University of Minnesota up until this point, where now he's an assistant professor there. He's published his work in many prestigious journals such as Science Advances, New Phytologist, American Journal of Botany, and he's received multiple NSF awards. So he's got lots of accolades here. And Daniel's someone who I think really does some incredibly innovative work. He takes an ecophysiological approach to better understand how exactly things like bryophytes and lichens work out there in nature, how they are able to live and persist in these extreme environments, and how they're responding to a changing world. I'm really excited Daniel's able to join us today, and I'm looking forward to learning more about his work. And thanks, everyone.
Thank you, Matt, and thanks for setting a bar that now I have to try to rise up to meet. My name is Daniel Stanton. As I said, I'm going to talk about some of the work that we're doing in my lab, trying to understand lichen symbioses and how they interact with changing climates. My goal for today is try to keep it, like, I'm going to try to, it's going to sound detailed, but like give you a broad overview of some of the way we're trying to frame our understanding. And a lot of this can be flagging as much as anything the gaps of what we don't know yet and where we're trying to go. But to briefly get started, I'll dig into more details later, but just because it's a broad audience, I'll acknowledge what is a lichen is one of these seemingly simple questions that is deeply existential and complicated and unending. But for getting started, if you're not used to it, these are the key things to just sort of keep in mind. There's at least one fungus, at least one photosynthetic thing. It can be green algae or cyanobacteria, or in a few rare cases, basically a seaweed, and other stuff. And then this very general combination of things forms a novel sort of emergent structure that operates sort of like an independent organism. And these things have many different origins in terms of the fungal lineages, in terms of the algal and cyanobacterial lineages. There's a lot of different forms. This is just sort of a small smattering of like the diversity of what falls under the category of lichen. And associated with all these origins and forms, there's a lot of different ecologies, habitats that they're in, whether it's on rocks in Antarctica or on leaves in tropical forests. There's a huge diversity that's encompassed in this. And it's important to keep in mind as we talk through this, because typically when we talk about lichens, we picture something that looks like this, or maybe that looks like these, and we're often forgetting just how wide that diversity of stuff is. And that's one of the areas that really interests me and a lot of the folks in my group. So, but the key piece of this that emerges on this, these are small. They're small combinations of organisms, or compared to a tree, to whatnot, and they typically don't have roots. They're not like a flowering plant that's drawing its resources from the soil and in close interaction with that. A lot of the resource exchange that they have is with the atmosphere. These lichens, there's various species growing on this trunk here, are all deriving a lot of their nutrition, but also their water sources, their interactions closely coupled with the atmosphere. And that means the chemistry of the atmosphere, the climate conditions of the atmosphere. And that's what gives them a lot of really interesting attributes that are the sort of the focus of the talk today. And one of those is that for a really long time, since like the second half of the 1800s, they've been used as indicators of air quality, because anything that's in this close nutritional exchange with the atmosphere is going to be taking up what it needs, but is also going to be directly exposed to a lot of pollutants. And people like William Nylander in Finland in the like 1860s, 1870s was already describing the fact that in heavily industrialized cities, you saw less lichens and less abundance and less diversity than in others. This has been really a continued effort that people work on to understand this, because air quality matters not just to the lichens, but it's associated with a lot of human health. And having easy indicators of that can be really effective. And this is work that continues to today. Dr. Natalia Koch in my lab has been doing some really exciting work around urban areas of Minnesota on this. And I'm not going to talk about it in a whole lot of detail, but if you wanted to get in touch with her, she's been doing transplants, she's been looking at what's growing there, and really trying to continue pushing forward our understanding of lichens as indicators of air quality in terms of pollution. And there's like this sort of long sort of history that we continue to build on. But today I want to focus on sort of a second area of aspects of interacting with the atmosphere that is of growing interest, which is thinking about climate as well. And historically, maybe initially, this was a challenge for people wanting to work on air quality, that they had to account for the fact that climate also affects the lichens. But now it's becoming increasingly maybe one of the ongoing uses of lichens as early indicators of what's going on in the air in terms of climate and not just pollution, especially as air quality in our urban areas in North America and Europe, for example, has been getting better. And there has been work by people like Bob Smith trying to map out vulnerability of lichens to climate and figure out whether there are specific species or combinations of species that are going to show different vulnerabilities to changes, whether it's changes in temperature or changes in moisture, and some aspects that we might see clearer effects with lichens that are decoupled from what's stored in the soil than we would with some other organisms. But the challenge is, how do we know what makes them vulnerable and how do they relate to climate? And this is sort of where it moves into some of the areas that I've been digging in myself. Because a lot of our knowledge for this, like those figures I was showing you just before, derives from basically distribution climate combinations. So we know how climate is distributed across a landscape, maybe Norway, maybe somewhere else. And we can look at where different lichen species occur at present, or maybe occurred in the past, and we can relate where they occur to the climates that they occur in, and forecast if those climates change, this is likely how those lichens would change. There have been some long-term and some shorter-term experiments and observations, but there's relatively few of these. And for the most part, this is very correlational. We can say this is where the climates, how these climates are today, and that maybe that drives somewhat where those lichens occur. But for being able to predict how they'll respond in the future, especially as climates move into what we think of as novel climates, combinations of temperature and humidity that we don't currently see near those places, then we need to make sure that it's not just based on these correlations, and we try to build in some mechanistic understanding of what's going on. What is it about that climate that the lichens are interacting with? What is driving them to be present and thriving, or dying out, or hanging on but doing poorly? And this is one of the areas where we've been trying to dig into, just sort of understand what drives that and underpins these big patterns, so that when we build predictive models, or others build predictive models, we can build them based on the right factors and not maybe ones that might not affect the lichens. And so a lot of today I want to spend on a couple of different projects that we've done in the lab focused on this organism here, Avernia mesomorpha. and use those kind of as a case study, because it's one that we've been using as, by chance we sort of stumbled into it, but it's one of our model organisms at this point for exploring these ideas. This led us into some deeper understandings, but things that we want to broaden out to a whole broader set of organisms. And so this Auvernia mesomorpha, if you want the common name, it's often called oak moss, which is a very annoying common name because it's not a moss. And on top of that, where we are in Minnesota, it mostly doesn't grow on oaks. It mostly is growing a lot of times in spruce and large tamarack bogs. And so it's not that it can't grow on oaks, but it's just, yeah, but that's the common name. You can think of it as oak moss as long as you can remember that it's not a moss. And so, but it's very distinctive and helpful to work with. And before I launch into describing any of the details, I want to be really clear that anytime I'm talking about work, it's only slightly stuff that I've done, and it's really the effort of a whole lot of other people who have made this possible. And there's a lot of different folks who work with my lab in different projects at different times. The ones that are underlined are ones that have contributed to the stuff I'm talking about today in some ways. And that's what I really want to highlight. And especially Abigail Meyer, who's a current PhD student, and Dr. Natalia Kosh, who's a postdoc, have led a lot of the papers that we're talking about today. Maria Valentin, Jean Pengra, Lana Levitis, Dennis Such, Lindsay Quam, Anya Nilis have all contributed to some of the efforts in the data collection, and some of them are on some of these publications. And then outside of the university, Rob Smith, who used to be at the Forest Service, Matt and Tammy McDonald, who's down the road from us in Minnesota at St. Catharines, have been really big parts of this too. And I just want to be really clear that this is never just me. I never operate, I can't do this all myself. I just get to be the one embarrassing myself up front here. And then the other piece of acknowledging context is not just the people, but the spaces in which we work. And so we're based at University of Minnesota. Minnesota is basically overlaps a whole lot with Minnesota Makoche, which is part of the Oceti-Sakowin Seven Councils Fire of the Lakota nations. It's also basically a confluence of a bunch of different things. It's the Twin Cities are established at Butte, which is a sacred site to some Dakota. It's the confluence of the Minnesota and Mississippi rivers. It's also it's been for a long time a meeting point of Anishinaabe and Dakota peoples, as well as other groups. And this area is the meeting point of 3 of the major biomes of North America. And so we're at liminal confluence spaces where the boreal zone meets the eastern deciduous meets southern prairie. And so all of these being at an edge and these meeting points also influences parts of how we think about things and what we're looking at as questions. So the work that we sort of got us started on this was a chance to collaborate with this massive experiment that's been established by the Department of Energy in central Minnesota. It's this kind of insane infrastructure in which to study effects of climate change on northern peatlands, they built these open top chambers that encompass multiple entire spruce trees in the middle of a bog. This is a bog here, these are boardwalks because it's all sphagnum bog. And then across in these chambers, they're either leaving it with regular CO2 or doubling the CO2 level, and they're applying warming, both underground and to the air. It's an insane operation that provides like a really cool opportunity to look at some dimensions of climate change. They're forcing some extreme ones, but it's a really powerful structure that we have a couple hours away. And back in the before they set it up, as they were installing it, Rob Smith and some other collaborators set up an experiment to look at the effects that these warming and CO2 treatments might have on lichens. And so they took Auvernia mesomorpha, this oak moss, and transplanted it into each of these different enclosures back in 2013, 2 years before the heat was even turned on. So they were able to give them time to establish and to get a baseline before they started the effects of sort of simulating climate change. And then we're able to track what happened with temperature and with CO2. And they recorded every year, they would go out there and recollect the ones that had been installed and weigh them and see how they changed. And so this is their key figure, that this 0 bar here would mean that anything that sits on that, the lichen hasn't changed in mass from 1 year to the next. It hasn't grown, it hasn't lost mass. You'd expect, like by chance, every once in a while a piece will fall off. There's going to be some noise to that. But what's really striking is that at low temperatures, at their control and at just a little bit of warming, there's been some growth, but none of the warmer temperatures show any growth. In fact, all of those warmer temperature conditions, the lichens have suffered and lost mass. And it didn't happen in the previous years until they turned on the heat. So they know it's not the transplantation, it's not the handling, it's that specific experiment. And so this is some really cool work that came out in 2018, and that inspired us to really want to dig in to bring sort of our tools as physiologists, as ecophysiologists into this, to try to understand what underlies that pattern. We're seeing a change, but what's driving it? There was some indication that they're getting bleached. So these are ones from different levels of conditions. So there's a change in how they look. We want to understand, like, are the symbionts, are the algae and fungi that make it up surviving this? Are they dying? Are there differences in terms of how they're doing physiologically? And one of the key striking things back here is that there's two, you can see there's sort of like 2 bars, one representing each sort of within each of these temperatures. These are 2 different CO2 levels. There's no effect of CO2 on what goes on. But we thought, well, maybe that's just at this really coarse metric of how much does a lichen weigh. But if we dig into its photosynthesis, its respiration, there might be some more subtle effects that are still significant that would accumulate over time, but that we're just not seeing with this really coarse metric. And so this was initially started by Maria Valentin, an undergraduate in the lab, and then built on by Abigail Meyer. So we went out in February of 2020 to collect, both because it was a good timing in terms of the school semester, but also because we could go out and collect when things were frozen. And Minnesota lichens are happy with freezing. They live through these conditions regularly. And that gave us this really amazing ability that we could collect all this material frozen, bring it back to the lab frozen, keep it in a freezer, and just pull it out, thaw it and rehydrate it, and know that it was in a good physiological shape. Most organisms you don't get to do that with, but we can do that. And so we could pull it out, and we tested whether this was valid, but we could pull it out months later or when it was convenient to run the experiments and do measurements on it. And you can see the effects of warming, that these chambers are really changing the conditions inside compared to outside. And there's a lot of details we could dig into. I'm going to give you like the brief overview of the results from this. But firstly, when we look at the change in mass, those changes, there's still no CO2 effect. And that effect of warming really continues to be present. Only at the control and the low temperatures were those lichens growing at all. And everything else was losing mass and in fact in worse shape than it was a few years earlier. There's been no reversal of things. It's mostly just sort of confirming what's going on, but then we can dig into some of the details. And we sort of bring a bunch of tools at this. The first one is just sort of looking at where is there chlorophyll, which is going to be indicating what goes on with the algae, and chlorophyll that's in sort of a functional shape. It's not just, is it green, but is it able to respond to pulses of light? Is it able to kind of have a functional response to things? And that's what this figure here shows up. And so what you can see is we've got this plate, we've got a bunch of different lichens that have been collected from the different conditions of chambers, different warming levels. And when we look at the activity that's going on in terms of the chlorophyll, the algal activity, only some of them light up. Some of these whole lichens, there's just nothing to be seen. In fact, there's brighter signals coming off of algae that happen to be growing on their label than there is from the lichen itself. And so, yeah, that's why these are marked. But you can see it's a nice way to visualize and see really clearly that there's been a loss of any photosynthetic ability from a bunch of these. And we can do this, I don't think I have the figure, but we've got this quantified as well. And you can see it with data if you'd prefer to see it with data. Sometimes the visuals are just more compelling than another figure. And this carries over when we look at other metrics. So that just shows us photosynthesis in terms of where there's chlorophyll present. But what really goes on with photosynthesis, or one of the things we're interested in with photosynthesis, is this key process of taking carbon dioxide out of the air and moving it into organisms' tissues and being able so that it can be used. So that process gets called carbon assimilation. It's one of the components of photosynthesis. And so we can look at what goes on with carbon gain, and it looks like what we might have expected from these results so far. There's carbon gain at low temperatures. The ones that were kept at not warmed, basically, or minimally warmed, are able to gain carbon still. The rest of them basically show no activity in terms of gaining carbon. And we can compare that to what's going on with carbon losses. And the ones at low temperatures are losing carbon. The ones at the highest temperature, highest warming, really probably are pretty much dead and inert. They're not gaining or losing. But I want to flag something here that's really intriguing that we've been trying to dig into. There's also an asymmetry. It's not just that there's growth or potential for growth in carbon gain with minimal warming and no ability to do that at higher warming, but there's this middle range where there seems to be a direct loss of the ability to gain carbon, but there's still carbon being released. And so there's some asymmetry going on between these two different things that's driving for maybe a continued loss of carbon. And I want to take that to really dig us deeper into what is going on with the lichen thallus. Because we often, and I fall for this, we all fall for this, we tend to treat them like an organism because they look like an organism. Their organismality is on messy philosophical ground, but we've got sort of an object that we can just kind of take and treat like it's a unit. But it's never just a unit. It's just the same as we aren't quite just human cells. We have all these other organisms within us that change our nutrition, that change our health, that change our physiologies. And this is even more so the case with lichens. So here's a nice illustration that Abby Glauser and some others, who was a former lab member, have drawn up to sort of illustrate an idealized lichen. And there's a lot of variation, but the key things is we've got these fungal components, but interwoven in that are algae that are doing the photosynthesis. There, in some cases, are cyanobacteria as well, or instead of the algae. There's all these other bacteria and yeasts and other organisms that are all hanging out in there as well. So there's a whole bunch of different things that are in there. And there's no reason why we should assume that they're all going to do things in coordination or respond together. And that's really one of the pieces that I think is what we need to be disentangling as we move forward towards understanding how lichens are going to respond, currently respond, or going to respond to future climates, is we need to figure out when do we have to do the messier thing of recognizing these component parts and engage with them, because they're all separate organisms. And so as separate organisms that have different evolutionary histories, they have at the very least the potential to each respond separately to different environmental cues, to different stressors, to what's going on. And so we have to think about this, a situation like this, as not just what responds at this organism level, but also what might be driving things for the algae, for the fungi, for the other bacteria. And that combination is going to lead what's going on. And so with the Avernia, perhaps it's losing the algae, but that doesn't mean that the algae dying doesn't mean that the fungus couldn't be persisting, or that the other things that were living in there might that were neither fungus nor algae might not still be hanging out or developing into new spaces. And we need to be able to engage with that to be able to forecast things. And I want to come back to Auvergne mesomorpha and take this idea that they're both sort of an organism, but there's also all of these components that are independent, and dig into sort of my second piece of story on this. That's sort of the work that I've been really published last year that I've been really excited about to try to dig into. So we're coming back to oak moss, which is neither a moss nor, in this case, on oaks. And thinking about one of the interesting aspects to them. And this is that when a lot of lichens, it's been shown that they're in close interaction with the atmosphere to the extent where in some cases they don't even need liquid water to be able to become active. They can use water vapor from humid air to be able to activate fill their cells full of water and become active. And this is an illustration of that with the Auvergne. It was a phallus that was starting dry. We had it sitting in humid air, and you can see how even 10 minutes in, it's already starting to show some activity at the very tips. And we can see basically we're seeing the algae within this phallus become active. And by an hour, the entire phallus is active. And within 2 hours, it's at activity levels that are basically comparable to what it would have if you sprayed it with water. So it's just sucking up humid air and activating. And this has been known for a couple of decades to some extent, but not fully examined in terms of like, how does this relate to climate responses, to engagement with the environment? and also, this is just the alga. And this is where things get interesting. So if we look back, so here I'm showing you that this chlorophyll fluorescence, this way of looking at photosynthetic capacity, it's the same basically, whether they're fully hydrated or they've been sitting in vapor for a while. They're really reaching the same capacity for photosynthesis from this visualization method. The same is true if we measure the amount of carbon that they're able to take up. So the photosynthetic activity in terms of like pulling CO2 out of the air into the lichen thallus. This is what they do in vapor on this x-axis, and here on the y-axis is if you took the exact same lichen and sprayed it with water and got it to optimal water content. And this line through here is not the statistical fit to that. That's the 1-to-1 line. That's just assuming if we just drew a line where there was equal amounts of these, that's that. It's almost identical. It's basically in humid air, they are able to reach the exact same rates of photosynthesis of carbon uptake that they would with optimal hydration. This is impressive. It's not a common thing for organisms to be able to just suck water from the air and do it. But when we look at carbon losses, it's very different. These red dots are, if we put it in the dark and we look at carbon, the rates at which carbon is being lost under those same conditions. And you'll notice that we're no longer sitting almost exactly on this line. Instead, we're falling way off of it, where the amount of carbon that's lost with humid air is about 40 to 50% of what it would be at sort of maximum hydration. So in other words, if you spray it with water, it loses twice as much carbon as if it's just hanging out in vapor. So this is intriguing because this sets up a real asymmetry between gains of carbon and losses of carbon depending on your water source. And this plays out across a bunch of temperatures, but it has some really big impacts that start to build up. So what we're seeing here, there's a lot of technical graphs, but these are what's called light response curves. So we're looking at how the amount of carbon, net amount of carbon gained, changes with the light level, and we're looking at it at different temperatures. So to walk you through one of these, so in the dark, we don't see carbon gains because photosynthesis can't happen. We're just seeing the amount of carbon that is being lost. And then as we raise the amount of light, the amount of photosynthesis that can happen, increases until it reaches a maximum and saturates. And these are really kind of common things. And what we're looking at here is two curves for each of these. We're looking at what's going on with liquid, if they're sprayed with water and sort of optimally hydrated, or if they're just hanging out in humid air. And for the most part, at lower temperatures, they're not that different from each other. There's some carbon losses. The carbon losses are bigger than the gains, but overall, things are tending to be not drastically different. But once we get up to the high temperatures, something really striking happens. These at 30 degrees Celsius, so at sort of like in the 80s Fahrenheit, we reach a situation where giving the lichen liquid water means that it is unable to break even in terms of carbon. More carbon is being lost, even that it is able to take up through photosynthesis, even in the best of light conditions. And so, and it's really marginal at 25 degrees Celsius. And what's important in this is this is just like instantaneous. This doesn't even account for what's going on at night. It sets up this threshold. We're past a temperature of 25 degrees Celsius, 77 Fahrenheit. It is unable to break even under the best conditions in the middle and in the course of the day. Under the optimal light, it's still losing more than it's gaining. And because this isn't even accounting for the fact that most of the time during the day isn't optimal light. There's periods of night where there's not enough light, there's no light. So there's always going to be periods where you're losing more carbon anyway. This is going to set for a really key threshold for when things are just going to be in deficit for carbon, especially in summer conditions. And what's neat about this is that this threshold here, this point where basically getting wet is a recipe for running out of carbon and going into deficit, is pretty closely aligned with where we see this lichen species occurring. If we map out for Vernia mesomorph, we go to herbarium records and we track down, all right, where is it found? Where has it been collected? What is the peak summer temperature that happens at those sites? What's the average summer temperature that happens at those sites? Basically, anywhere where the average summer temperature gets above this threshold, we're no longer seeing that occurring. This seems to be pretty closely related to where its climate distribution range is. We don't know that this is the mechanism, but it's certainly suggestive that this is one of the things that's driving it. And I suspect that even in the areas, the southern parts of its range where we find it, we almost always find it in bogs. We find it in places where it's going to be in high humidity conditions that allow it to do a whole bunch of carbon gain at limited loss, not areas where it's reliant on rain for gaining activity. And it sets up presumably predictions for what may go on into the future to try to understand things, because it's going to be this balance of how often does it get wet at high temperatures that may drive where it does well. And this is something that's been seen with some mosses and stuff. There's this really counterintuitive idea that what gets a number of these organisms through hot summers is staying dry, not getting wet. Because if you give them water, they're actually in these really dangerous conditions where they lose more carbon than they can gain, rather than you would think that getting some extra water through the summer is how you eat your way through. And so getting at this piece here helps us understand what might drive that climate difference, maybe about these conditions and specific thresholds. So how do we explain this? Well, we know, as I said, like we got to think about this nuance. These are separate organisms. They may be closely intertwined, forming a new structure, but that doesn't change that they're still separate organisms with different biological histories, different biologies, different underlying biochemistries. And so we've known for a while that the fungi and algae do seem to take up water at least semi-independently. There's some interaction if you put water if you add liquid water to one end, and the fungal hyphae will wick it across, and that will wet the algae. They're not fully independent, but there's definitely operating differently. And there was some work back in the 90s where they looked at just a few electron micrographs, and you can see these algal cells pumping full of, so going from dry to relatively plump and active, if not completely so, in high humidity, and maybe less so in what's going on with the fungal hyphae. So there's some sense that they're responding differently to these things, and they're kind of decoupling their response. And we wanted to dig in, Abigail, among others, wanted to dig into this more deeply and understand, okay, so we're seeing this at this coarse physiological level, but we're not actually tracking what goes on with that organism. When we see respiration, see carbon lost out of the thallus, all we know is that out of this whole cluster of all these different organisms, carbon is being lost. We don't know who's losing it. And it's really hard to figure out who is losing it because they're all intertwined. We can't separate them out and track that. And so we wanted to like sort of get it, if we want to get at what's going on and who is active and who's inactive, we've got to maybe use some other methods. And she was really interested in trying to do some transcriptomics. So we'll talk through transcriptional activity. And if you're not used to seeing these plots, what this is looking at is basically in maybe hopefully not embarrassingly crude version, but simplified version of this. We're taking our sample, grinding up, and we're looking at the RNA that's being expressed at a given moment. So this is not exactly, it's one of the ways of trying to get at what's going on in terms of the intracellular biochemistry and activity. It's not what it's doing with the proteins, but it's what it's trying to set itself up to do. And so a measure of activity. And to understand what's going on with this, we typically, one of the ways we use are what are called volcano plots, because sometimes they're 90 degrees and they look like a volcano. The big thing is that there's a whole lot of transcripts. Each of these dots is a different sequence of RNA that's hanging out in there. And we're comparing two conditions. So in this case, we're looking at, for example, when they've been hanging out in humid air compared to when they've been given liquid water. And we want to see, okay, there's always going to be some slight differences in how many copies of this transcript there are, how many copies there are of this one. But we want to know if those differences are significant or not. And so there's a bunch of statistical thresholds for this. And this is what this is trying to show. Each one of these dots is a differentially expressed transcript, but the ones that are colored are the ones that are standing out statistically in terms of being really way more of them in one condition than the other. And it can be that there's more when they're dry than when they're wet than when they're in vapor, or it could be that there's less when they're wet than in vapor. But the big thing is they're different. So if we look at this for our Evernia, for our lichens, we see something really striking. This is not what we were expecting in some ways. If you look at the fungus, it's kind of straightforward. There's definitely some differences in what's going on in terms of its transcriptional activity between being liquid, hydrated, and vapor. So it's changing. It's responding to being given that other water source. And that aligns with the respiration. Although when we look at the identity of these transcripts, there's no really killer story that we can tell. We don't want to there's a whole bunch of different transcripts. We want to take like 80 things and make up a story as to what those might mean. But we had some ideas as to, is it something that's directly tied to carbon metabolism that we can make a clean story for? No, there's no sort of straightforward story, but there's a difference there. But the algae don't change at all. And at first I was like, oh, well, of course, because we see the same level of photosynthesis going on, whether it's humid or they're being sprayed with water. So of course they're not going to respond to that. But the more we thought about it, the more this is really kind of striking, because it means that within this organism or this thing that's like an organism, that's this closely associated thing, we're seeing a change in the fungus. We're seeing a major change in respiration, big changes in what's going on for one of the main component organisms. And the other one just doesn't care. It's hit its level of activity and it's just doing its own thing completely separate from what the fungus is. And this is not how we tend to think of these lichens, because it's way more convenient to treat them as a unified pseudo-organism that is going to respond in a coordinated way. And that may sometimes be true, but not always. And so this is a really striking shift. And as I said, lichens are more than just these two primary components. So we try to look for some of the others as well. And there's a yeast that's often found in the outer layers of lichens. It's a Basidiomycete yeast. We were curious to see, does that one, what is that one doing? It's doing the same as the alga. It's not responding to these big changes. The bacteria are, but interpreting bacteria stuff's a little bit messier. So we don't know for sure that this doubling of the carbon losses is exclusively coming from the fungus. It might be coming from bacteria as well. It could be, but it's certainly not coming from this other fungus, nor is it coming from the algae. There's a bunch of nuance there that we really need to dig into further to understand this. So to quickly summarize things, the key thing here is that these component organisms that make up a lichen are activating differently. They're responding in a really kind of decoupled way to environmental cues. Maybe not always, but certainly under some sort of key moments. And this decoupling isn't just a neat academic detail. It certainly seems to affect physiology and long-term carbon balance. And it might explain what's going on with climate tolerances and ranges. So this is a really neat piece to disentangle. It makes my life harder to have to think about these different scales, but it's really fascinating. So to move towards the last sort of phase of this, I want to think about, like, how common is this? So we did this with this workhorse, lichen, because that's what Rob happened to have used in his experiments. It's a cool one. We're right in Minnesota. We're at one of the southern edges of its limit. And so we're right at a range limit, which is kind of practical. But one of the things I really tried to push in my introduction, and we really try to be really conscientious about in our lab, is that lichens are tremendously diverse. And the vast majority of what we know about lichen physiology and ecology has been biased towards large lichens that are easy to work with equipment that was designed for plants, so large lichens that are easy to work with, and ones that are living in temperate to high latitudes that are close to where the researchers are, or in Antarctica, because Antarctica is kind of sexy and it's a place to go, and it has big showy lichens. But that's not where a lot of the diversity is. There's a huge amount more than just this one major origin. And lichens have evolved many different times. There's different reconstructions of this, but this is one estimated phylogeny. And then, so this is the fungal orders that show lichens, and then intersecting with different algal and some bacterial orders. You can see there's a whole lot of different combinations that occur. And yes, some of them are really common. These are sort of the best studied ones here, but there's a lot of diversity there. And so we know that diversity can matter. There's some evidence that at coarse levels, the type of photosynthetic component, of photobionts, we call it, changes how they respond to humid air. It changes how they tolerate water saturation. We know that there's a bunch of differences between these different algae that in fact interact with climate. Natalia has done some work on this. So we know there should be differences that are driven by that diversity. And so Natalia really set out this is her first project when she started her postdoc, to try to expand the range of what's going on with this. And it'd be too expensive to do transcriptomes on all of them, but we can at least look for this asymmetry, what's going on between photosynthesis and respiration. Are the different components, what we think are different components, responding differently to different water sources? And so we've tried to obtain as many different lineages as we could easily get our hands on of fresh material to work on. And so we took a couple collecting trips to Mississippi to get subtropical stuff that just makes it into the southern US, but are lineages that are found throughout the tropics and more dominant there. There's one or two Mediterranean ones from California and a few like straight up tropical lineages from Puerto Rico to complement that. And so overall, we managed to get 41 different combinations of algal and fungal origins out of the 70 to 80 that are known, depending on who's counting. I don't want to get into the arguments exactly how many there are, but we've got quite a few different ones covering 14 fungal orders, 6 photobiont orders, and so it's a good smattering of that diversity. If we look at it on this figure, this sort of like cross section, the stars are all the ones that we managed to get specimens to represent. So it's not perfect coverage. There's gaps. I want this, but this is also just kind of, we don't know what it is. Chlorococcoid is a vague one, but it's a chance to really kind of broaden out our perspective to make sure that when we're trying to make claims, we're not just making it extrapolating off of oak moss and claiming this is how lichen overall might work. And to give you like the super summarized version, it's a really widespread phenomenon. At least this asymmetry, this kind of decoupling in what's going on. The way to see it is across this is a tree phylogeny of the fungal components. We have a couple of different growth forms and types of substrate and algal physiologies coded here. And the darker blue this is, the bigger the difference in what's going on with the alga from what's going on with the fungus in terms of like its ability to respond to bait. We puzzled over how to build this index into like a simple number for a while, but basically the darker blue it is, the more this is at play. If it goes red, it means that there's maybe more respiration going on in humid air than there is photosynthesis. And really, blue shows up everywhere, across all these different lineages, across all these different conditions. It's a really widespread phenomenon, with the exception of this cluster here, that are all associated with cyanobacteria, and specifically Nosta. Even some of the cyanobacterial ones show a little bit of that. So the key thing here to take away is that this seems to be a widespread phenomenon. Now we need to dig into like, how does this relate across different temperatures? How does it relate to climate ranges? There's a lot more to be done to break this up, but we think it's pretty confident that it's not just that one. It's not just something that oak moss does that's unusual, but rather something that we need to be building into our mindset when we think about lichens going forward. All right, so lastly, if we think about this, these lichens are messy. We've had to sort of engage with the fact that it's a whole bunch of component organisms, but we name them off of the primary fungus, and that's our name. We say Auvernia mesomorpha. That name comes from the fungus, but what we're talking about is this emergent combination of components. And one of the things that makes that interesting is that because we're naming that emergent combination, to some extent, the identities of those components can change. And as long as it still looks like the same emergent thing, and it still has a closely related enough fungus, we still treat it as the same thing. And so this is an added piece of nuance that we're having to sort of engage with, not just us, but a lot of lichenologists. There's a nice framing of this kind of idea that came out of Doolittle and Booth that Toby Spavilla brought my attention to, that you can have these changing components in the same overall emergent unit, as the idea of like, what we're talking about is the song, not the singers. The song can retain its identity even as the singers that do it swap over or change. We can have the same emergent structure that we might call a lichen with two different genotypes, strains of fungus associated with the same alga, or we can have one fungus associating with one alga genotype or species, or it depends on how, or a different one, or maybe both of them together. And so and yet we still have what we will treat as the same unit, but presumably if there's differences either in the algae or in the fungi, this is going to lead to differences in their climate responses, in their underlying biologies, or they might. And there's growing evidence for this within a single lichen species. So based on the fungus, we can see not only that there'll be different algal strains associated with it, but which strain you find can vary with climate. It might be that some strains are found in cooler climates and others are associated with the same fungus, but in warmer conditions. This is at big geographical scales, but there's also been work in Germany showing these variations going on as you just go up a mountain within the same species. So all these pink ones are one species of lichen. These yellow ones are an overlapping, but closely related species. And as you move up the mountain from basically Mediterranean up into cold climates, you see that the relative abundances of different algal strains changes both within a species, and then as we swap over to different species, changes again with those. So there's all this variation going on in terms of what those components are in response to climate. And we actually see evidence that it's maybe even happening relatively fast in response to climate. When Lima Lulevicius and Abigail analyzed those Avernia mesomorphas that I talked about in this climate experiment, and we looked at the ones that had been sitting out in the hotter temperatures, they had different clades, different strains of algae dominating within those. So there's a shift there that maybe happened. We don't know what drove it, but there's a shift there. And with Natalia's work with lichen transplants is like the one unpublished bit that we're still finishing up. It looks like we're seeing some shifts within a year as we put them out into new conditions. And this is one of the really big open questions to me as we dig into this stuff with lichenology, lichens and lichen ecologies. How often do these things change? How often do we see change turnover? Also, maybe with the fungal component, we see what might look like a continuous thallus can have components trading in and out within it. We have no idea how fast or frequently. I think I talked to people and I have heard from very well-informed guesses a full spectrum from, no, of course they don't change very fast at all. They're the same to, oh, they do this seasonally. And we just don't know. And a bunch of us have been trying to figure out how do we actually track individual cells and work these numbers out? Because it's going to be a really key thing to trying to understand how they might change. Because if this is associated with how they respond to climates, this may also be one of the paths towards understanding how they will respond to future climates and their ability to adapt. One of the reasons to think about this is that we know, and there's been more work on similar dynamics that go on with corals, which I think of as lichens of the sea. There are comparable thing there where we see in response to climate shocks, there's changes in the symbionts that are present in sort of a coral association. And there's actually been some really recent work that's showing this, trying to actually induce better climate adaptation by bringing in new symbiodinium strains, new symbiont strains that are adapted to warmer temperatures and getting the coral to take them up. And it's still in lab conditions, but they're able to get them to basically swap out, not just observe them doing it, but like induce resilience to future climates, to warming through swapping out those symbionts. And so understanding whether this is a possibility for lichens, not maybe as a conservation intervention, but to understand what their potential is, seems really valuable and interesting. So this is one of the sort of wrap up, running out of time, think about sort of what we want to take from here. So one of these is we saw this with Avernia mesomorpha. I showed you that like this broad asymmetry is present across a whole lot of different lichens. We still don't know really what's going on with the temperature thresholds. There's lichens that grow in warm, hot, wet climates. How do they do it? It's clearly not all operating with the same stresses as the oak moss, or we wouldn't see lichens everywhere else down into the tropics. So we need to figure out what those thresholds are, what those differences are, and it opens up a whole range to understand that. Does this help us understand climate ranges? It looked really compelling with oak, with Ivernia mesomorpha, but that may not be true. It's not going to be the only factor explaining things for maybe tropical lichens. What else controls those ranges? But this may be part of the picture forward. And then lastly, does this change in component organisms start to become not just a way of describing how they change with climate, but offer a way for them to buffer or adapt or adjust? And if so, how fast does it happen? Is it able to happen at any kind of useful rate to respond to changing climates? So to sort of finalize and wrap up, I want some takeaways. If you care about lichen monitoring for air quality and whatnot, then there's still some good things, right? Like lichens reflect unique aspects of air quality and climate, and they're valuable for that. And the better we can understand this and how they respond to climate, the better we can use them in sort of sophisticated ways as predictors or early sentinels of how climate is changing. Because we want to be able to know what exactly they're responding to be able to talk about what is changing. And some of these aspects, their traits, seem to be applicable not just to one or two lichens, but there's some generalities that emerge from this. And Natalia and Matt and others we're working on this to try to figure out broader frameworks so that we can generalize, because we don't want to have to do detailed studies for each species that we want to use as an indicator. That's just not practical. It's really especially not practical in places that have less resources for that. Or if you care about the lichens, as I do, there's a bunch of interesting things going on. There's this tantalizing possibility of partner change and these component changes. There's this idea of asynchronies in activity, the value, and then these bits of thinking about these integrations of like organism as partially organisms and partially not, and how we move forward takes thinking about that evolutionary context, this physiology, and a lot of other things. And so with that, I will wrap up. And sorry if I didn't leave much time for questions. Thank you.
Thanks very much, Daniel. Really appreciate your perspective to help convey how fascinating and complex these iconic symbioses are, and just how much we have to learn yet too. So
Trying to make it like
yeah.
I'm an ecologist because I love complexity.
Yeah.
So I get excited about the nuances and the complexity, but it's also part of the goal is to find where can we generalize and not just get stuck in the weeds.
Sure.
Yeah.
Sure. All right. Thank you. I think we have time for a couple questions. Going from Greg here.
Great stuff. I've never understood how active the fungus is at assimilating their photobionts, or is it just they take up what's available? Because I was just wondering if whether that photobiont community is changing because of climate, and therefore that's what gets in the lichen. Are they actively, kind of a community assembly question?
They seem to do quite a bit of selection. There's, and Matt can talk about this more than I can. There's certainly like specific species of fungus, populations of fungus will be sometimes quite closely associated with one or a couple line lineages of algae. In other cases, it's much more mixed, and you can get multiple lineages within one. But there seems to be varying, but definitely a fair bit of selection. They're not just taking any random alga out there.
Yeah. I was wondering though, like
Yeah.
this Trabuxia, do they just pick up the Trabuxia that happens to be there? Or are they selecting particular whatever? So I know
What do you
there's,
think?
I think that's the question, and that's I know
Yeah.
Daniel's been working on something very similar with a different set of algae, Trinipolia algae, and trying to see are these.
Mosaic, it's a really good question, and we don't know. Yeah.
Any other questions? Yes, Anderson.
Hey, Daniel, thank you very much for the talk. I got curious, I don't know if you mentioned in a loss, try to interpret in the graphs, but is there a reason why taking from the air humidity comparing to the, like think about the rain, air seems to be a better source of humidity or water than grain itself?
I don't think, I think better is unclear. I think it's just, it's an available source. Water follows osmotic gradients. And so if there is an osmotic gradient from humid air into the cell, then it will move in. I think what it opens up for these organisms, because they're small, they're thin, it's a lot more windows for hydration. They're what we call poikilohydric. They don't control their water content particularly well. That's certainly not like mammals or vascular plants that tend to keep a constant hydration. Instead, they dry out and rehydrate. And so being able to take up moist air just creates much longer windows of activity that are available.
Great. I think we have time for just one more question.
How well were you able to kind of track like humidity within those chambers and like because they almost work like a greenhouse a little bit. I mean, they do have an open top, but like, do you think future experiments would like try to control humidity as another factor?
You mean the big climate change experiment chambers?
Yeah.
They track humidity. The humidity is one of the big challenges with it because the way that you mix the air for CO2, they have to have fans blowing and mixing it up. Otherwise, it doesn't diffuse fast enough. And so because there's all this airflow, they tend to be drier than the surrounding environment. And some of the impacts that we see that look like warming are probably a combination of warming and drying. And in fact, a lot of like warming and drying, because warmer air causes them to dry faster in the first place, that's an added, there's a lot of conflation in a lot of experiments of what's going on with warming and drying. So it is a challenge to it. And I think future experiments will need to try to take that into account or find ways to do that. It's just the logistics and engineering involved in even getting what they did is amazing. I think yeah. Does that address your question enough? Yeah. The rest of our stuff's been done on much smaller scales where we control the humidity, but it's different.
Cool. All right. Thanks very much, Daniel. I really appreciate it. Thanks everyone for coming out. See you next week.
Yeah.